Hello, welcome to the In Vivo Imaging Success Knowledge Sharing Video Forum. I'm Alexandra Delille and Director of Technical Applications at PerkinElmer for In Vivo Imaging. Today we will discuss the best luciferase reporter combinations for in vivo dual reporter assays. If you have questions and want to explore further, feel free to email me at alexandra.delille@perkinelmer.com. Here you can see a list of luciferase reporter genes. Their respective substrates, emission wavelengths, and size, as well as cofactors, are also outlined. Firefly and click beetle luciferase are both ATP and oxygen dependent, rendering these reporters a live-dead stain. Only when the cells are alive, they produce ATP and light emission will occur. Unlike Firefly, Ranella and Gaussian luciferase do not require a cellular energy source. The solentrazine substrate itself provides enough energy. Nanoluc is a new, small luciferase from Promega, but both the enzyme and its optimized substrate, furimazine, have not yet been validated in vivo. Bacterial luciferase and its applications will be discussed in a future video. A reference table for popular codon-optimized luciferase constructs. Codon-optimized luciferases result in higher expression and therefore stronger signals. Namely, the DNA codon sequence is synthetically humanized, and further modifications to both the vector backbone as well as the cDNA, such as removal of peroxisomal targeting sequences and glycosylation sites, internal restriction sites, etc., result in improved stability and dramatically higher expression in mammalian cells. The substrate D-luciferin. D-luciferin is a chemical substance found in bioluminescent organisms. D-luciferin is the substrate for various luciferases, and its oxidation in the presence of ATP generates photon emission. Synthetic D-luciferin can be purchased as a water-soluble potassium or sodium salt. Simple intraperitoneal injection allows for absorption and systemic biodistribution. D-luciferin is a small molecule that freely crosses the cell membrane. D-luciferin does not cause toxicity or an immune response. D-luciferin is not metabolized, but simply cleared by the kidneys. D-luciferin kinetics. Since D-luciferin biodistribution is fairly rapid, and the kinetic curve may be tissue-dependent, it is important to assess the kinetic curve and optimal imaging time point post-injection for each model. More information on methods to determine the kinetics of your substrate can be found in the Basics of Optical Imaging Part 2 Kinetics and Animals video. The substrate solentrazine. Solentrazine, or solenterate luciferin, is the substrate for a number of marine bioluminescent enzymes such as Ranella and Gaussian luciferase that catalyze solentrazine oxidation to produce light without the need for ATP. Native solentrazine and synthetic derivatives are commercially available. Native solentrazine is preferred for Gaussian luciferase. Ranella luciferase can also catalyze solentrazine H. Solentrazine dissolves in organic solvents. Recently, both a red-eject solution from PerkinElmer as well as a water-soluble solentrazine from Nanolite were released. Solentrazine is prone to auto-oxidation and therefore may autoluminesce. The Viviren substrate from Promega has been modified to protect oxidation sites. Solentrazine kinetics. Because of the fast enzymatic flash kinetics, intravenous injection is recommended, with image acquisition shortly after injection. Keep in mind that solentrazine is a substrate for the MDR1 transporter and is rapidly exported from high PGP expressing cell lines and the blood-brain barrier. Remember that reporters that excite and emit above 600 nanometers will maximize sensitivity, as the absorption by hemoglobin is significantly reduced in this part of the spectrum. Therefore, Firefly and click beetle red offer higher sensitivity versus Ranella and Gaussian. Good dual reporter combinations. Firefly and Rhinella luciferase is the oldest pair. They can be discriminated by both wavelength as well as substrate. Although Rhinella emits in the blue, it is still a good reporter and can be used at depth. The click beetle red Gaussian combo. In this case, a truncated, non-secreted Gaussian is utilized as native Gaussian luciferase is secreted. Again, these can be discriminated by both wavelength and substrate. Click beetle red has a narrower bandwidth in comparison with Firefly luciferase and therefore may be a better option when it comes to spectral unmixing. The click beetle red click beetle green combo. These luciferases use the same easy-to-use substrate D-luciferin and are spectrally unmixed by wavelength. Emission spectra. It is important to be aware of the emission profiles of the respective luciferases. Gaussian and Rhinella can be discriminated by both wavelength and by sequential substrate injection from Firefly and click beetle red luciferase. However, click beetle green and click beetle red both utilize D-luciferin as their substrate. As shown in the graphs from Dr. Zawa and colleagues, click beetle green and Rhinella have a red emission tail. Therefore, one has to be cautious. While the click beetle red click beetle green combo is great in a subcutaneous model, in deep tissue, the lower wavelength light emission of click beetle green is mostly absorbed, and the surface radiance will consist of a red tail which cannot be discriminated from the click beetle red emission spectrum. In conclusion, for deep tissue models, Gaussian or Rhinella versus Firefly or click beetle red are preferred, and sequential substrate imaging is highly recommended. Examples from the literature. This is one of the earliest dual reporter in vivo publications by Drs. Wang and Alderi, illustrating tumor cells with SV40 constitutive Rhinella luciferase expression and P53 response element-driven Firefly luciferase expression. As you can see, adenovirus E6 effectively degrades P53, decreasing Firefly luciferase light emission. Adenovirus like Z serves as a negative control. Another example is published more recently by Dr. Mezzanotte and colleagues. In this case, sensitive dual-color in vivo bioluminescence imaging is accomplished with a new red codon-optimized Firefly luciferase and a green click beetle luciferase. Labeled cells were injected subcutaneously, and spectral unmixing of the signals was determined in the IVIS spectrum. Unmixed and composite images were generated. Click beetle green is pseudocolored in green, while PPY RE8 is represented in red. Mixed populations result in yellow. Another elegant study by Drs. Na and colleagues, as published in Blood, illustrates a dual bioluminescent reporter system allowing non-invasive concomitant imaging of T-cell trafficking, expansion, and activation of nuclear factor-activated T-cells in vivo. A truncated Gaussian luciferase is constitutively expressed, while click beetle red expression is driven under control of the NFAT promoter. NFAT activation plays an important role in T-cell activation and T-cell development. NFAT signaling and development into CD4 or CD8 single positive cells could only be detected in the thymus of female recipients, indicating either absence of positive selection or prompt depletion of double positive thymocytes in the male recipients. Red effect lentiviral particles to create your own luciferase cell line with confidence. PerkinElmer recently launched 3 ready-to-use lentiviral particles. The first one encodes the red Italian Firefly luciferase emitting at 620 nanometers. The second lenti co-expresses the red Italian Firefly luciferase and GFP, facilitating immunohistopath and flow cytometry. Keep in mind, however, that GFP attenuates Firefly luciferase about tenfold. And lastly, a new and very bright Rhinella luciferase is featured that emits at 535 nanometers. This Rhinella luciferase is an excellent choice for dual reporter assays with an inducible Firefly luciferase reporter. Each of the constructs drive expression of the reporter gene under ubiquitin C promoter, the best choice for stable expression. The image illustrates and compares signal intensity of a lung metastases model, with cells expressing either vanilla luciferase, as seen in the left mouse, or firefly luciferase, as seen in the right mouse. Thanks for viewing this knowledge sharing presentation. Stay tuned for more videos. If you have questions, feel free to contact me by email at alexandra.delille@perkinelmer.com.